In this video, I'll go over some of the fundamental concepts and processes integral to using MyAssays. An assay is an analytical test used to determine the presence, concentration, or enzymatic rate of targeted entities. These targeted entities are called analytes. An example of an assay is a salivary cortisol ELISA. This assay is for the quantification of the analyte cortisol in salivary samples. Some assays are developed in-house, meaning the laboratory has created the assay from scratch, but many assays are commercially available. The manufacturer creates and sells the assay in a kitted format where the lab personnel will have everything they need to perform the assay. Inside the kit, lab personnel will find all reagents, plates, standards, QC, and a kit insert or manual detailing how to perform the assay and how to analyze the data. There are different types of assays. Some return values while others return positive or negative results. The assays that use a calibration curve comprised of standards with known values to interpolate the concentrations of unknown samples are quantitative assays because they are able to quantify the concentration of an analyte in a sample. Assays that use samples with known characteristics to identify thresholds denoting positive or negative results are qualitative assays. You may also encounter semi-quantitative assays. These assays may report the values in arbitrary units that coincide with a qualitative scale. For example, 0 to 7 might be characterized as negative, 7.1 to 10 as equivocal, greater than 10 as positive. This way you'll have a result, but not a concentration. And by using this result, you can also assign the samples a qualitative result. Our salivary cortisol example is a quantitative assay. As you can see, the standard curve is used to interpolate the concentration of various unknowns. Standards are specialized samples that have a known concentration. The corresponding raw measurements associated with these standards can be used like a ruler to determine the concentration of unknown samples. There are three main laboratory processes that take place during the performance of an assay: sample preparation, data measurement, and data analysis. MyAssays is designed specifically for in vitro analysis, meaning that assays are performed on samples that are collected and prepared in a lab rather than in a living organism. Sample types can include blood, urine, saliva, and tissue samples. These samples are often manually pipetted into tubes or microplates. A microplate can contain a variable number of wells. The most common formats are 12 by 8 96 well plates and 24 by 16 384 well microplates. This nomenclature refers to the number of columns and rows on each plate. You can see here a cartoon representation of a 96 well microplate. If the assay is pipetted into tubes or vials, those will be arranged into a rack layout. Racks contain multiple tubes. A common layout is a 10 or 20 tube rack. You also have the opportunity to define custom microplates or rack numbers within the software. If you find your data analysis doesn't look as expected, one of the first steps of troubleshooting should be to check the plate layout and the sample preparation. Reagents are any compounds that are used to complete the assay. In commercially available kits, these will be provided from the manufacturer. Typical ELISA reagents could include items like wash buffers, antiserum, assay buffer, enzyme conjugate, TMB, and stop solution. Once the kit protocol has been completed, the raw measurement will be obtained from the reader or detector, imported into MyAssays, and analyzed. There are different types of instrumentation and detection technologies used to measure assays. MyAssays can support data measured using different detection technologies from different instrument manufacturers. A plate reader is a common tool that is used to measure ELISAs, or enzyme-linked immunosorbent assays. Plate readers will measure absorbance or optical density, and this correlates to the amount of analyte present in the sample. Luminometers are specialized plate readers that are used for luminescence immunoassays, or LIAs. Luminometers measure RLUs, or relative light units. Similar to ELISAs, in LIAs, the RLUs correlate to the amount of analyte present. LIA microplates have the same layout as EIA plates, but they differ in that ELISA plates are clear while LIA plates are opaque. Radioimmunoassay, or RIA, is another methodology used to measure assays. A radioactive isotope like iodine-125 is used to generate counts per minute, or CPMs. RIAs are different from ELISAs and LIAs in that these assays are commonly performed in tubes or vials as opposed to in a plated format. Gamma counters are the instruments that are used to read RIAs. After measuring the raw data for an assay, the next step is to perform the data analysis. In order to analyze the data, we'll work in MyAssays Desktop. Protocols are MyAssays files that contain all the information for data import, analysis, reporting, and export for each assay. These are normally created in accordance to literature, such as a kit insert, and will perform the data analysis as described. All of the files associated with MyAssays will be stored in the Assay Data root folder, which you can access from File, Data, and clicking on this link. There you'll find the subfolders automatically created by MyAssays. Alternatively, you can access your protocols by clicking on the Protocol tab on the left-hand side of the MyAssays Desktop Explorer. Within the protocol, there are several tools to help you complete your analysis, first being the Data Import Wizard. This wizard will walk you through the process of importing your data, automatically recognizing many common formats, and stepping you through the process of choosing how to import your data if the format is not automatically recognized. You can select what metadata to import and choose the naming convention as well as the file location, and even configure the software to automatically import, analyze, and export your data when a file is detected in a specified folder using folder pull. You can import not only your raw measurement data, but also any additional parameters pertaining to the data, including sample IDs, standard concentrations, dilution factors, and plate IDs. There are different types of measurement data. Endpoint data is a single measurement that's performed after a fixed incubation period. You can see in this example data one measurement for each position on a 96-well plate. Kinetic data is a series of measurements performed over fixed time intervals, most commonly used for enzymatic assays. In this example, you can see the position listed on the left-hand side, along with multiple measurements corresponding to different time points listed on the side. In this chart, you can see the multiple data points as they're plotted over time in seconds. Spectral data consists of a series of measurements at multiple wavelengths. Again, in this data, you can see the position is listed on the left, along with the multiple measurements corresponding to different wavelengths out to the side. Again, in this chart, you'll see the multiple measurements plotted on a scale of wavelength in nanometers. Multiplex data is a single plate of measurement data that is used to quantify multiple analytes from a single assay run. In this example, you can see a measurement for each position for each analyte, and upon analysis, this data will be used to interpolate concentrations for the multiple analytes. After your raw measurement data has been imported into the software, you can click Calculate to analyze the data. This will execute the specified data analysis based on the parameters described in each individual protocol. Once your results have been calculated, you can view the report under the Results tab and see the various export options under the Report tab. You can open in Excel, Word, or export as a PDF. Or you can click on the Home tab and click Export to open the Export Wizard and explore the additional export options, including custom options there. Under Properties, you'll find tabs that allow you to specify the data analysis and organize and format your data. Under the Matrices tab, we can see our matrices. Each matrix holds a certain kind of data. For example, raw data, blank corrected, concentration, and %CV. These matrices are referenced, and with the exception of the raw data matrix, they're created by transforms. For example, after I have initially entered my raw data, I'll have only a raw data matrix. Then I can use my Transform tab to perform a blank correction. I'll set the input matrix to be raw, and I'll create the output matrix of blank corrected. Then I can add another transform, for example, the standard curve fit transform. I can use my blank corrected data as the input matrix and create the concentration as my output matrix. I can build out my data analysis using this matrix transform method and format how the data will appear in my Matrices tab. Here we can specify any numerical formatting, the report contents, sample labeling, as well as adding value bars. Additional report formatting can be found under the Report tab by clicking on the report element and making any changes needed here. I've covered a lot of topics very briefly in this overview. For more information, please see the subsequent videos on MyAssays familiarization, as well as data import and export, folder pull, transforms, matrices, as well as rack and plate layouts. If you have specific questions, please feel free to contact a member of the MyAssays support team.